morning, good afternoon, good evening, everyone. My name is Lucilia Morias. I am the Scientific Program Manager for the Cell Therapy Tracking, Circulation and Safety Committee, or TRACS at HESI. I'd like to take just 2 or 3 minutes before we start our program and handing it over to Bill, our moderator, to give you a few housekeeping items and reminders very briefly. First, I'd like to thank all of the speakers, meeting moderator for their participation today, our collaborators at TRACS for their support in co-organizing this meeting, TRACS members in the planning committee of this event, as well as all of you in the audience for joining us wherever you might be in the globe. So here's a few housekeeping items. As you may have heard at the beginning of the meeting, this webinar is being recorded, and if you miss anything at any point, you can go back to it. We'll have a recording available for viewing later. You are welcome to turn your cameras on if desired, and all the participants are automatically muted, but you can communicate with us via the chat box or the raise hand function, but please keep in mind to remain muted throughout the meeting if not speaking. We will have questions at the end of all 3 speakers' presentations during the live 30 minutes discussion. And for that, we will use Slido to address questions from the audience. So at that point on, from now on, you can start going to slido.com using your computer, a cell phone, or a tablet, and enter the code that you see here on the screen, 868-602, and all the questions will be captured. During the discussions, we will have the screen on the shared with all, so you can follow the questions as well. When entering questions, please indicate the speaker's name if your question is addressed to someone specifically. And now it's my pleasure to introduce Dr. Bill Shingleton, who will open this meeting and will also moderate the live discussion session at the end of the presentations. Dr. Bill Shingleton is Director of Applications and Biology for Cell and Gene Therapy at Cytiva, and is also the co-chair for HESI TRACS Committee since 2016. Bill is a biological scientist by training with a background in inflammation, biology of aging and connective tissues, and applied his research in the fields of autoimmune, degenerative disease, and oncology. He has experience both in academia at the universities of Cambridge and Newcastle upon Tyne, and industry, having integrated roles at Unilever, Research and Development, GE Healthcare, Life Sciences, and now Cytiva. Dr. Shingleton's current role is focused on leading a global team of biologists and engineers developing technology to enable the development of tools and processes essential for the manufacture of cell and gene therapies. And with that, Bill, I'll hand it off to you.
Thank you very much, Lucilia. Welcome everyone to this webinar. Hold on a sec while I share my screen, which should now be visible to you guys. Is that the case? Someone could nod so they can see my screen?
Yes.
Yes. Okay, brilliant. Thank you very much. Cool. So start off with just want to give a brief introduction to HESI and the TRACS committee and what we've been working on since the committee formed. So HESI is an independent scientific not-for-profit organization recognized as a leader in bringing together multi-stakeholder, multidisciplinary teams of international experts to solve scientific challenges around risk assessment and safety. It's a neutral platform, it's transparent and totally independent. You can find more information about what this transparency and the work that they do by visiting the HESI websites. But what their aim to do here is to ensure that we can avoid any kind of conflicts of interest, which enables the participants to join these committees and work for the greater good. HESI committees maintain a public-private balance, and we do not conduct any lobbying activities, and we do not take and make any policy recommendations. So we operate internationally, and by that we mean that HESI board members and participating committee members are from all over the world. We try to address collective challenges that are not bound by a single geographical area. Outreach and training activities are conducted in all corners of the globe. So we achieve our mission in a variety of ways. There are three components of the HESI model: convene, translate, and protect. By creating a collaborative environment where scientists from academia, government, industry, and non-government organizations can come together. Our programs encourage the development of meaningful studies that ask the right questions, structure and framework, and develop solutions that can inform decision-making. Implementation: the outputs of the programs are easily transferred from laboratory to journal page. In real life are utilized to enhance human and environmental health at both local, national, and of course our international levels. So HESI is a connector, bringing together those various elements, looking to solve scientific challenges around risk assessment and safety, and facilitating collaboration across academia, government, industry, and NGO scientists. Our committees produce impactful, evidence-based science, better for informed decisions. The third point is around translation. So translational nature of HESI's activities is the key goal, is to actually get this out there so people can see and use and put the knowledge and information into practice. Okay, so cell therapy, tracking, circulation, CT TRACS. What is this committee all about? So there are various committees within the HESI community, or HESI family, as you like, and this is how the work is delivered around certain or into specific areas or specific topics. There are currently 15 active committees and other initiatives, each with several subcommittees working in particular focus areas. CT TRACS is one of the youngest of the 15. It was formed in late 2015 and started its activities in 2016. And our mission was to facilitate, as you can read it from here, to facilitate and fill the gaps in development tools and gaps in knowledge that we perceive to be required for development and progression of cell-based therapies. These slides, I'm sure, will be shared so you can read the detail in your own time. We are inclusive of all cell types and all kinds of cell-based therapies. CT TRACS leadership, as you'd expect from HESI, is broad, diverse, and truly global. Co-chaired by Tineke van der Hoorn from the MED in the Netherlands and myself. We have committees around point of administration and tumorigenicity. Point of administration biodistribution work group is chaired by Brooke Helfer and Vladimir Ponomarov. And the tumorigenicity work group by Charlotte de Wolf from the MED and Hirota Bando from Shorendenko in Japan. The committee is supported by Lucilia, who you heard from already today, and Connie Chen. And without the input and direction and coordination of our staff, of the HESI staff, these committees and indeed the work that they do to coordinate, convene, and translate cannot be possible. So, cell therapy. So, CT TRACS. What are we about? What's our purpose, if you like? Well, the strapline is securing the safety of cell therapies to realize their potential. Our mission is to facilitate the translation of cell-based therapies to the clinic by driving the development of tools, methods, knowledge required to evaluate the safety and fate of therapeutic cells. When we formed the committee back in late 2015, I was working for a bit then at GE Healthcare that was focused on imaging, and with an interest and previous career in cell therapy, wanted to really try and see what we could do to leverage this technology. Talking to people in the industry, we were soon connected with HESI and identified that this is a gap. So how cell therapies and indeed gene therapies are, but cell therapies in particular, are so different from other pharmaceutical agents that the tools and technologies to evaluate their safety do not necessarily exist, or we are begging and borrowing from other elements that make it difficult to actually assess the safety of these cell therapy products. So we formed two committees or two subgroups within that. One to look at the point of administration and biodistribution. How do we know where these cells go? What do they do? How long do they persist? Do they survive? Do they proliferate? Where do they locate to within the body, and do they stay there? The tumorigenicity. With the types of cells mainly focused in the regenerative space, regenerative medicine space, how do we know that these cells are not going to inappropriately replicate in the wrong place in the body and cause tumor in the patient? And predicting assays and being able to tell whether or not a cell therapy is safe from this perspective. Tools to do this are not anywhere near as well developed as the tools that are available for, say, small molecules or biologics. And this is what this committee is all about and trying to do. Since we've been formed, we've been busy not only developing tools within our individual organizations and such like, but we've been pulling together articles, and we have two publications out on the topics, one from the point of administration biodistribution group and one from the tumorigenicity group, outline a case for what we are trying to achieve as a team and the current status within the industry. Our group, as you can see from this slide, is indeed truly global and truly representative of those areas that that were mentioned earlier on in the introduction, in the terms of NGOs and consortia, the top left, academic across the top, our government regulatory bodies, and then we have our industry partners in the bottom on the right hand side. We also have some people, we're advancing automatically, excuse me. We have new teams hopefully joining us, or new organizations hopefully joining us in this year in the shape of Charles River, EMS, and we hope to welcome Broken Strings to our teams as well. So, with further ado, I shall now move on to introduce our first speaker. Tineka's biography. So our first speaker is my co-chair of TRACS, Tineka van der Hoorn. Tineka works for the Medicines Evaluation Board as a preclinical assessor for ATMPs and cell and gene therapies at the MED in the Netherlands. Tineka has an MSc in biology from Utrecht and a PhD in tumor biology from Leiden University, and is currently the regulator at the Dutch Medicines Evaluation Board. Tineka has been on the CT-TRACS committee for a number of years now and has been co-chair since 2019. From this point, I welcome Tineka, or Tineka's recording, to be played. Thank you very much.
Good morning, good afternoon, good evening. My name is Tineka van der Hoorn. I'm non-clinical assessor at the Dutch Medicine Evaluation Board, and I work also as an assessor for the EMA. Today I'm going to present you some regulatory considerations for the preclinical development of cell-based products and present you some learnings for the future. I would like to emphasize that what I'm going to tell you today is a personal opinion, especially as the field is still advancing and also in the member states in Europe, slight different opinions may exist. With regard to the cell-based products and the non-clinical development, few aspects have to be noted. Cell-based products are a diverse group of products. They might differ in their cell origin, being either from autologous or allogeneic origin, might differ in their modification status, being either unmodified, which are called cell therapies, or genetically modified cells, which are called gene therapies, and they also may be different in the differentiation state, ranging from terminal differentiated cells to undifferentiated stem-like cells. They are also variable with regard to the type of indication. They're developed either for monogenetic disorders, for which also very young children are treated, or they're developed for late-stage cancer, or another group is otherwise, among which also fall the products for which are aging related. One common thing holds true for all products. The non-clinical development program does not follow a tick box approach that concerns the aspects of pharmacology, pharmacokinetics, and toxicology, which is not standard. The last 15 years, guidelines have been published from the EMA, but also lately from the ICH on the non-clinical development of cell-based products. Some overarching guidelines are the risk-based approach guideline and the guideline on quality non-clinical and clinical requirements for investigational HMPs. Specific for unmodified cells is the human cell-based medicinal product guideline, and guidelines that are applicable for gene-modified cells are the non-clinical studies required before first clinical use of the gene therapy medicinal products, or the quality non-clinical and clinical aspects of medicinal products containing genetically modified cells. And very lately, the ICH came together to write a guideline on non-clinical biodistribution studies for gene therapy products. However, we consider that the best guideline to be followed for these type of products is THINK, so only 5 letters. And an example for non-clinical development, one should ask, why will the developed product be efficacious for the chosen indication? What is already known from similar type of products, and which risks needs to be approached non-clinically? So for this approach, one should consider the product type, the indication, the investigational nature of the product, and also the clinical experience with similar type of products. This approach is based on risks, so risk-based, and follows a case-by-case approach. During the past decades, development programs for cell-based products have been evaluated via scientific advice procedures or via marketing application procedures. Well, this presentation will show you some learnings from the scientific advice procedures on cell-based therapy products and some learnings from marketing application procedures of cell-based products. Starting off with the learnings from scientific advices on cell-based products. So we set out to investigate the development program of cell-based products via reading scientific advice reports and the information that the applicant provided. We retrieved that from the EMA database on scientific advices. There were 218 advices that concern 153 products. Of those, 94 were cell-based and 86 were human cell-based products that had also non-clinical information in their package. Well, there was a large variety of products, so we tried to classify the products based on their cell origin, either being allogenic or autologous, and based on their genetic modification status, either not or genetically modified. We analyzed, amongst others, which type of studies were performed in vivo. So we found out that most studies were done to provide information on the proof of concept. Then in vivo studies were done to find out about the safety of the product. Afterwards, most studies were done to assess biodistribution, and least in vivo studies were done to assess the tumorigenicity. However, we could not see a clear role for the classification in this analysis for the type of in vivo animal studies. We also did analyze the type of biodistribution and tumorigenicity studies that were done, and a little bit more on techniques or design of studies that were used. This is all described in the paper that came out recently, of which the information is at the bottom of the page, which is published in the British Journal of Clinical Pharmacology. In the next slide, I would like to present a little more on the biodistribution studies, as this is also of relevance for this presentation. As you can see, at the top of the page is a table which shows whether studies were available, either already performed or planned or requested, or whether they need to be justified in red, and whether it was agreed that the study was not present or whether it was not mentioned at all, so probably not relevant. As you can see, there are more cases for the autologous not genetically modified cells in which it was agreed that it was not relevant to study biodistribution, as these are most not necessary or regarded not mentioned. Whereas for the allogenic or the genetically modified cells, it's more often agreed that applicants performed a study or that a study even was requested. At the bottom, you see a figure where we present how the applicants analyzed the cell persistence. You see that in the majority of the cases, persistence was analyzed. Migration was for the majority of the products evaluated in target and non-target tissues, and for some autologous products, only target tissues were evaluated. With regard to the animal model that was used for these in vivo biodistribution studies, the majority of the cases, the human product was tested in an immune-compromised animals. However, also quite some products were tested in either diseased or healthy mice, and in that case, the animal equivalent cell of the human cell was used. With regard to the method, that seemed to be rather standard methods that were chosen by the companies, because only 3 biodistribution studies analyzed the presence of the cell with PET, MRI, SPECT or BLI, methods that are more advanced and also can address dynamics. So what we conclude in our research is that non-clinical in vivo animal studies were mostly evaluating the proof of concept of a product. With regard to tumorigenicity, we found out that an in vitro approach only may suffice, and the omission of in vivo studies appear to be possible for products with sufficient clinical experiments. It was also clear from this research that the choice for in vitro and in vivo studies and the study design was determined by the type of the product, the state-of-the-art knowledge and clinical experience with similar type of products. Apparently, the development programs are very diverse, so one size does not fit all. And it's considered very relevant that a dialogue between developers and regulators starts as early as possible in the process. The question also arose whether there is a more tailored guidance possible for similar type of cell-based therapies. So then we studied European public assessment reports of authorized products, authorized cell-based products. So we grouped these type of cell-based products in three categories: either CAR T cells, of which there are now 4 authorized, or gene-modified CD34 positive cells with a positive opinion, which are also 4 currently at the European market, and a class of unmodified cells. However, the unmodified cells, cell-based therapies, appear to be too different, also in their development, to provide kind of common rules for assessment. So this group will be excluded from this analysis. So the coming slides, I will show you what we came across in similarities on the e-parts from these different products. So starting off with CAR T cells. CAR T cells are a group, are specific T cells, but this analysis may be relevant for gene-modified immune cells for oncologic indications. So with regard to the pharmacology, the in vitro aspects that are most often studied are the vector copy number, the cell viability of the CAR T cell, the expression of the target, the affinity and the specificity for the target, the cytotoxicity of the CAR T cell, the ability to proliferate, the type and the extent of the cytokine production, and the effector versus memory T cell ratio. In vivo is a bit more difficult. We have an immune cell. Studying that in an animal will be impossible unless you have an immune compromised animal, and as the efficacy also requires an immune system, you should actually make use of an immune compromised humanized mice if you will test the human CAR T cells. And of course you need a xenotransplant that should be fighted. Sometimes an animal equivalent of the human cell is generated. We call this a surrogate product, and this can then be tested in the mice itself. For pharmacokinetics, which is also non-standard for cell-based products, applicants can study biodistribution. However, this appears less often done for CAR T cells that are against blood-borne tumors, but this may become likely relevant for CAR T cells against solid tumors. In case the biodistribution is studied, the analysis method is rather standard. Mostly PCR, immunohistochemistry, or imaging can be used. With regard to the safety, the safety data from pharmacology studies and toxicology studies is regarded relevant to discuss the safety, and the toxicity study, if that is done with a clinical product, then it's done in an immune-compromised animal model, which has, of course, the limitations for the translation to human. What's also seen is the tissue cross-reactivity study, which might occur ex vivo or in silico, and it's important to see whether there are no off-targets. The in vitro tumorigenicity study that is done is most of the times an in vitro immortalization assay or an IL-2 independent growth assay. Often the insertion site analysis studies are conducted within the clinical studies. For the CD34 positive T cells, positive cells that are transduced with an retroviral vector or an lentiviral vector that encodes the transgene, the following development program headlines can be given. Most of the times the in vitro aspects that are studied are the vector copy number, the expression level of the transgene, the percentage of positive cells, and the stem cell mass of the product, and that means how many early and late progenitors, the lineage, and also the differentiation profile that can, if that still is functional. With regard to in vivo, most of the times an animal equivalent of the human cell is also transduced with the lentiviral or retroviral vector encoding the transgene, and that is then given to the animal that reflects also the disease. So the disease animal, most of the times this is a disease animal model, is the mice. With regard to pharmacokinetics, these dossiers most of the times contain a biodistribution study, and they analyze the migration to an engraftment in several tissues. And also the persistence of the cells is analyzed, and this occurs both in heterologous and homologous models. What is measured is the vector copy number and also the transgene expression, and these are both evaluated by means of PCR, immunohistochemistry as more standard methods, and sometimes also companies explore imaging by transducing the cells with a lentiviral vector within GFP as a transgene to see where the cells end up. However, this is steady state analysis and not dynamic. With regard to the toxicology, safety data from the pharmacology is regarded relevant, but also toxicology, dedicated toxicology studies are done with the clinical product in an immune-compromised animal model. With regard to tumorigenicity, the in vitro insertion site analysis is very important. In vitro immortalization assay is also often presented. With regard to the in vivo tumorigenicity studying, sometimes if a long-term study is done, tumorigenicity endpoints are taken along in these studies and can be regarded as relevant. With regard to reproduction toxicity, often a weight of evidence approach is followed. So to conclude and summarize, a more tailored guidance based on groups of similar products for cell-based therapies could be an option. For CAR-T, we see mostly in vitro studies. In vivo proof of concept is used often in humanized mice models. With regard to the biodistribution, this study is not always included, and for safety, the evaluation in animals has limitations, and the tumorigenic risks often is addressed clinically. With regard to gene-modified cells, CD34 gene-modified cells, the PD is both in vitro and in vivo studies, and these are important for proof of concept and providing clinical treatment rationale, and also help estimating the first in human dose. With regard to the pharmacokinetics, the biodistribution study supports both the pharmacology and safety. And with regard to safety, there is evaluation in both homologous and heterologous animal models, and the tumorigenic risk that is addressed both non-clinical and clinical. For reproductive toxicity, it's often a weight of the evidence approach. An early dialogue between developers and regulators remains valuable, but how is that possible within the European Union? Well, there is the Innovation Task Force for discussing the product. It's very informative for both parties, and it's not binding. There are national scientific advices that are more informal, pricing nationally determined, but not binding for the EU. The central scientific advice at the EMA is most often only written. It's binding for the EU, and there is reduced pricing for small medium enterprise. Then there are prime procedures at the EMA. That's only for promising products. The rapporteur is linked to a certain product already early in development phase. The marketing authorization procedure at the EMA is done in the following manner. Two rapporteurs look to the product and write an assessment report, one peer reviewer, and all other member states give comments to this procedure. This is the end of my presentation. I would like to thank you for listening, and I would like also to thank my colleagues for the helpful discussions on this topic. Thank you.
Apologies, just wrestling with the mute button on this one. Thank you very much, Tineke, for a fascinating presentation. Just to remind everybody in the webinar to place your questions via slido.com, and the code to enter the question once you get into slido.com is 868602. We have a question and answer session at the end of all three talks. So thank you very much again, Tineke. So our next presentation is going to be delivered by Kate Ziagi. Kate is the veterinary officer for or at the US FDA. Kate is a pharmacology and toxicology reviewer for the Office of Tissues and Advanced Therapies at the United States FDA. She's received her doctorate in veterinary medicine from the University of Pennsylvania with a focus on small animal clinical medicine. Prior to joining the FDA, she was a practicing veterinarian and medical director at a large veterinary hospital. Holds a master's degree in molecular genetics and a master's degree in cellular biology, developmental biology, and biophysics from Johns Hopkins University. Kate, thank you very much indeed. And now welcome the playing of Kate's presentation. Thank you, Courtney.
Hi there. My name is Kate Ziagi, and I work in the Office of Tissues and Advanced Therapies in the Center for Biologics Evaluation and Research at the U.S. Food and Drug Administration. Today I will be presenting on Preclinical Considerations for Cellular Therapies, a CBER FDA perspective. So today I will talk about the organizational structure of CBER OTAT and the products we regulate. I will talk about preclinical considerations for assessing the safety and activity of cell therapy products. I will talk about preclinical considerations for assessing cell fate after administration of the cell therapy product. And last, I will describe opportunities for early interaction with CBER OTAT. First, I'll talk about our organizational structure and the products we regulate. There are 3 product review offices in the Center for Biologics Evaluation and Research. I am in the Office of Tissues and Advanced Therapies. There is also an Office of Blood Research and Review, as well as an Office of Vaccines Research and Review. We regulate a wide variety of products in OTAT, including gene therapies, stem cell and stem cell derived products, functionally mature differentiated cells, products for xenotransplantation, therapeutic vaccines and other antigen-specific active immunotherapies, blood and plasma-derived products, tissues, devices, and combination products. Here are some examples of cell therapy products that we regulate. Mesenchymal stem cells, or MSCs, various cell-based therapeutic vaccines, so for example, dendritic cells or irradiated tumor cells, products comprised of exosomes or extracellular vesicles, induced pluripotent stem cells and embryonic stem cell-based products, and various immune cell-based products including NK cells, TILs or tumor-infiltrating lymphocytes, and other T-cell-based products. And it's important to note that T cells that have been genetically modified, like CAR T cells or TCR T cells, are considered gene therapy products in our office and are regulated as such. So some potential safety concerns for cell therapy products include changes in cell properties resulting from ex vivo manipulation, so for example, expansion, encapsulation, and scaffold seeding, potential immune response, tumorigenicity, inappropriate cell differentiation, so for example, ectopic tissue formation, cell distribution to non-target sites or tissues, and risks of the delivery procedure. So here, IND stands for Investigational New Drug Application. And as you can see in the graph, from 2016 to 2020, cell therapy INDs increased from 63 to 151, which is an increase of 140%. So interest in this field is rapidly growing. Next, I'll talk about considerations for preclinical programs for cell therapy products. So how does preclinical data contribute to the planned clinical trial? It provides a justification for a first-in-human clinical trial in the target patient population with the target disease. It supports the starting clinical dose level, dosing regimen, and route of administration. For the product administration procedure, it establishes feasibility and reasonable safety. It supports the inclusion and exclusion criteria in the clinical protocol, and it identifies potential toxicities and physiologic parameters to help guide clinical monitoring. A good starting point to understand how our office evaluates preclinical safety and activity is the Guidance for Industry, Preclinical Assessment of Investigational Cellular and Gene Therapy Products. So when thinking about developing a product, this is a very good resource for considerations for a preclinical testing program. In the guidance, there are recommendations for general program design, as well as recommendations for cell therapy products, gene therapy products, and therapeutic vaccines. As you saw from an earlier slide, there is a huge variety of products that come through our office, as well as a wide variety of cell therapy specific products. Because of this diversity, the ideal approach is a flexible, no one-size-fits-all review of these products. This is based on accumulated knowledge and experience and on available technology, and it is science-based and data-driven. When designing a preclinical testing program, sponsors should consider the mechanism of action and the intrinsic properties of the product, the indication for the product, the appropriate animal species and animal models that will provide relevant data to the questions being asked, and sponsors should also consider if there's any existing preclinical or clinical data for the clinical product or a similar product, the proposed patient population, or the clinical route of administration. Then that data can be evaluated for quality and applicability to the preclinical program. The main components of a preclinical testing program for cell therapy products are proof of concept or POC studies, which should demonstrate evidence of biological activity in animal models that recapitulate aspects of the clinical disease. Cell fate assessment, which consists of survival and engraftment, distribution, differentiation and integration, and tumorigenicity. These can all be affected by route of administration and the in vivo microenvironment. And these assessments help in the interpretation of product activity and safety. Immunogenicity and host response. Sponsors will often have to use immunocompromised rodents for human cell therapy products to avoid rejection, although sometimes sponsors will also use analogous animal products and assess immunogenicity in an immunocompetent host. It is important to keep in mind that ex vivo manipulation may change cell properties even on autologous cells that could result in an unexpected host response. Encapsulating or seeding cells also introduces additional materials that could result in a host response. And last but certainly not least is safety, which I will get into a little bit more in the next couple of slides. So when considering the design of preclinical studies, the sponsor should ensure that the studies are non-biased, so randomized with masked assessments, that the studies mimic the planned clinical scenario as closely as possible, so dosing regimen and frequency, route of administration, and the concomitant use of other products. Sponsors should ensure that the appropriate control groups are included. So for example, if immunosuppression and a monoclonal antibody are also given as part of the dosing regimen with the investigational cell therapy product, it is worth considering a vehicle group and a group with and without the monoclonal antibody. Sponsors should use a product that is identical to the clinical product to the extent feasible. So as just discussed, depending on the product, the use of an analogous product or surrogate product may be appropriate. And sponsors should evaluate multiple dose levels that bracket the proposed clinical dose level range. Sponsors should use adequate numbers of animals per group to enable robust study interpretation. And it's very important to mention that we strongly encourage incorporation of the 3 R's of animal testing when designing preclinical programs. The first is reduce, and this means the use of just a single species or the use of a single study to gather both pharmacological and toxicological data whenever practical, and the use of non-terminal evaluations instead of multiple cohorts of animals with terminal endpoints when justified. The second is refine, and this means the incorporation of pain management and humane endpoints and the use of non-terminal imaging modalities. And the last is replace, which means the replacement of selected animal studies with in vitro studies if such alternatives exist or can be developed, or replacing higher order species with lower order species. For example, using rodents in place of non-human primates if adequate data can be gained from the rodent studies. Studies should be of sufficient duration to assess both acute and long-term outcomes. They should incorporate multiple time points for evaluations, and the specific time points depend on the biology of the product. There should be comprehensive bioactivity, distribution, and safety assessments. Standard toxicology endpoints can include, but are not limited to, mortality, clinical observations, body weights, clinical pathology, and terminal assessments like gross pathology, organ weights, and histopathology. There can also be product-dependent endpoints like immune response, disease-dependent endpoints like survival and reversal of phenotype, and functional endpoints. And other specific assessments include cell fate assessments. So for example, PCR-based techniques, immunohistochemistry, and other modalities, which I will discuss next. So now I'll talk about preclinical considerations for assessing cell fate post-administration. The 4 main considerations when thinking about developing a preclinical testing program to assess cell fate include survival and engraftment, distribution, differentiation and integration, and tumorigenicity. So when assessing survival and engraftment, you're basically asking the question, how long do the cells persist? Viability and engraftment may be affected by the biocompatibility of the cell delivery device and the cell therapy product. So considerations include things like cell shearing and adsorption onto the walls of the catheter or syringe, the route of administration, the genetic relationship of the cells to the host animal, so whether the cells are autologous or syngeneic, allogeneic, or xenogenic, the immune status of the host animal, so immunocompromised versus immunocompetent, and the timing of cell administration relative to the onset of the disease or injury, so basically the pathophysiologic status of the microenvironment. So when assessing distribution, you're asking the question, where did the cells go? This will be influenced by the specific properties of the cell therapy product and by its route of administration. So there can be systemic versus localized distribution, distribution to targeted versus non-targeted tissue. The cell therapy product can also distribute proximal to sensitive tissues, so for example, neurologic or reproductive toxicity. And there are anatomic considerations, so for example, proliferation in enclosed spaces. When assessing differentiation and integration, you're asking the question, what happens to the cells? Is there continued differentiation? Is there dedifferentiation? Is there transdifferentiation? Is there integration into tissues or organs, which could potentially be anatomic or structural, or could be functional integration? Or is there ectopic tissue formation? And last, for tumorigenicity, you're asking the question, do the cells have tumorigenic potential? There are many factors that may influence tumorigenic potential assessment. So as mentioned in the previous slide, the differentiation status of the cell type in the product. So this can range from undifferentiated or embryonic to terminally differentiated or specialized. The extent of cell manipulation during the manufacturing process and the resulting growth profile. So for example, there could be minimal manipulation, culture expansion only, culture expansion with or without growth factors, ex vivo differentiation, or ex vivo transduction with or without cell expansion. Other factors influencing the tumorigenicity assessment are the expressed transgene of genetically modified cells. And for this, it is worth noting that, as I mentioned before, we consider genetically modified cell-based products to be gene therapy products. So for example, CAR T cells, and we regulate them as gene therapy products. For more information on this, I would recommend reading the preclinical guidance document on assessment of cell and gene therapy products that I mentioned earlier. And other factors include the potential to induce or enhance tumor formation from existing host malignant cells and the target population. As we've discussed, cell fate studies can give us a lot of information on survival and engraftment, distribution, differentiation and integration, and tumorigenicity. But they can also give additional information on safety and provide serial data over specific time intervals in the same animal, which can reduce animal use. They can give information on optimal dose level and optimal timing of administration and repeat administration. And they can provide information on a potential mechanism of action and can help bridge the preclinical data to the clinics by helping to inform trial design and appropriate monitoring for the proposed clinical trial. In addition, non-terminal preclinical imaging can provide a foundation for clinical imaging and inform feasibility of clinical imaging modalities, as well as inform safety parameters. When considering various imaging technologies for cell fate assessment studies, some ideal features would be that it provides data that is real-time, has the ability to do serial imaging over an appropriate time course, which may be a few months or maybe lesser or greater based on the specifics of the cell therapy product being evaluated, that it has the ability to quantify viable cells, that it has high sensitivity and specificity with a good signal-to-noise ratio, and that it is non-toxic to the cell product and the recipient animal model with minimal effects on cell function. So it is important to understand the effect of the manipulation on cell viability, phenotype, and activity. For example, genetic labeling or cell loading with contrast agent may affect cell viability, may affect phenotype, or may affect activity. Some examples of modalities to assess cell distribution include PCR-based assays, immunohistochemistry, optical imaging such as fluorescence or bioluminescence, MRI and nuclear imaging, and whole body acoustic imaging. Next, I'll briefly go over some considerations for selecting appropriate animal species and models for preclinical studies. CBER's recommendations for preclinical development of cell therapy products are flexible and are done on a case-by-case basis, depending on the specifics of the product, the route of administration, the disease indication, and other product-specific considerations. So there is no default to the use of non-human primates, and there is no default to the use of both a rodent and a non-rodent species. Sponsors should assess safety, distribution, and bioactivity using an appropriate animal model for each preclinical study. And sponsors should understand the limitations of the species and models used and provide scientific justification for their selected animal species or model of disease. Some important things to consider when selecting an animal species and model are the relevance to the target patient population, so the phenotype, pathophysiology, and clinical outcomes, the permissiveness to the cell product, so will the cell product be autologous, allogeneic, or xenogenic in the animal model, and should an immunosuppressed model be considered? And the route of administration and delivery device and procedure should reflect the proposed clinical route of administration and delivery device and procedure as feasible. If there are any differences between the preclinical study and the planned clinical trial, sponsors should provide a discussion of the potential impact on the translation of the animal data to humans. Other limitations and factors should be identified and discussed. Things like availability of animals and cost, size, sex, age, and housing needs, animal care concerns, technical feasibility, historical and baseline data, and statistical limitations, which often come into play when using large animal species, such as non-human primates. And as discussed previously with the 3 R's of animal testing, it is important to consider alternative testing to support animal studies. So in summary, the preclinical development program for a cell therapy product should be science-based, should enable translation of benefit to risk in humans, should be designed using the best available technology and methods to date, and should incorporate judicious use of animals. And last, I'll talk about opportunities for early interaction with CBER OTAT. This is a schematic of the life cycle of a cell therapy product as it moves through various stages from early development all the way to BLA, which is a biologics license application, and post-marketing. In the early stages, there are two types of meetings where we can provide feedback on preclinical testing programs. These are Interact meetings and Pre-IND meetings. So INTERACT stands for Initial Targeted Engagement for Regulatory Advice on CBER Products. The sponsor's goal is to obtain early feedback on a product development program for a novel investigational agent. It is a mechanism for early communication with OTAT where there will be non-binding, informal scientific discussions between CBER review disciplines and the sponsor. It is an initial targeted discussion of specific questions and issues that sponsors may have. OTAT provides non-binding written comments generally one day prior to the scheduled teleconference as feasible. And for Interact meetings, OTAT does not provide teleconference minutes. Requests for Interact meetings should be sent to the email address here, and below is the FDA website, which contains more information. As far as timing of when to request an Interact meeting, it should be when a sponsor has generated preliminary preclinical data, so some feasibility and POC data and maybe some safety data, but is not yet ready to conduct definitive preclinical safety studies. The advice we will provide consists of design of proof of concept or other pilot safety and biodistribution studies, adequacy of the selected animal species and models, acceptability of innovative preclinical testing strategies, products, or delivery modalities, and advice on modification of a preclinical program or study design as applicable to ensure judicious use of animals. So again, in the life cycle of a cell therapy product, an Interact meeting can be requested during the development stage as discussed, and a pre-IND meeting can be requested during the preclinical phase. The goal of a pre-IND meeting is to provide feedback to the sponsor to be able to achieve a successful IND submission. A pre-IND meeting is a non-binding, formal scientific discussion between all review disciplines and the sponsor. The review disciplines consist of a CMC reviewer, which is the chemistry and manufacturing controls reviewer, a pharmacology and toxicology reviewer, and a clinical reviewer. In addition to preclinical data and proposed pivotal studies, it is important to include any product characterization concerns as well as the proposed design of the planned clinical trial. And as touched on previously, a pre-IND meeting can be requested when proof of concept and preliminary safety studies have been completed and the sponsor is ready to conduct their definitive pivotal safety study. More specifically, the summary of all completed preclinical studies should include in vitro and in vivo studies, justification for animal species and models, study designs, and resulting data and interpretation. It should also include complete protocols for the proposed definitive safety, distribution, and POC studies. These protocols should contain detailed information on animal species and models, dose levels, dosing regimen and procedure, study endpoints, and sacrifice intervals. So in summary, CBER OTAT regulates a wide variety of products, including cell-based therapies. The preclinical program for any cell therapy product is not a one-size-fits-all and is determined on a case-by-case basis. Preclinical data submitted in the IND should support the safety and biological activity of the cell therapy product in the proposed clinical indication. Determination of the distribution and cell fate of the investigational cell therapy product following administration in animals is important for characterizing the product activity and safety profile. There are multiple opportunities to obtain FDA feedback on preclinical development plans prior to an IND submission, including Interact meetings and pre-IND meetings. And here's my contact information, as well as additional resources for any regulatory questions you may have. Thank you for your attention, and thank you so much for giving me the opportunity to share the CBER FDA perspective on preclinical considerations for cell therapies.
Okay, thank you very, very much indeed, Kate, for a really fascinating and thorough walkthrough of your work and your department's activities. Thank you very much indeed. So again, a reminder, as Cecilia just popped in the chat, slido.com with the code 868602 in order to enter your questions, which we will return to after our third speaker. Our third speaker is Marta Realuna, who is the Scientific Regulatory Affairs Director at Asphaleon. She will be presenting a case study with the NTRAC product, utilizing nanoparticles for cell tracking and lessons learned from the regulatory process in the EU. Marta has a pharmacy degree and a specialist postgraduate degree qualification in industrial pharmacy and galenic formulation. She has over 7 years experience in drug development in the pharma industry as an R&D project manager on the development of drug candidates and 6 years of experience in regulatory affairs. As Scientific and Regulatory Affairs Director at Asphaleon, Marta is responsible for scientific and medical writing and regulatory documents working in the regulatory strategy, regulatory roadmaps, ATMP designation, scientific advice and qualification of novel methodologies for biologicals, biomaterials, medical devices, and advanced therapies. Also manages the regulatory work package of Horizon 2020 projects. Marta, thank you very much, and we will now play your presentation.
Hello everyone, this is Marta Rayo speaking from Asphaleon, a scientific and regulatory affairs consultancy firm. However, today I'm talking in representation of the NTRAC consortium. I will present a case study, the NTRAC project, where we are utilizing nanoparticles for cell tracking, and I will focus the talk on the lessons learned from the regulatory process in the European Union. We will start with a brief introduction of the NTRAC project, and the NTRAC project is an Horizon 2020 EU-funded project. We will explain why regulatory guidance is interesting and relevant. We will talk about the classification of the product and the lessons learned from the regulatory process followed, concluding on the main regulatory and non-clinical issues to be resolved. The NTRAC project aims to develop a sensitive multimodal nanoimaging agent capable of providing safe body monitoring administered living stem cells. Our goal is the evaluation of cells migration and biodistribution patterns in order to provide early assessment of cells therapy effectiveness. We have already synthesized 2 prototypes of nanoparticles for structural and functional tracking of stem cells, specifically as a therapy on muscle regeneration. Nanoparticles can be detectable by magnetic resonance due to its iron oxide core in one case, and also by computed tomography scanning in the other one, due to its external gold layer. Within the execution of the H2020 project, we aim to demonstrate that cells maintain their therapeutic properties after labeling, and to assess the safety of the product, including GLP compliance preclinical toxicology studies. In terms of functionality, the labeled cells with the NTRAC nanoparticles are being injected in vivo into a surgically induced skeletal muscle injury of a sheep model, to be then tracked by non-invasive imaging technologies. Finally, we aim to demonstrate that tracking can lead to computational predictive models for early assessment of treatment effectiveness. Machine learning algorithms will be used to increase imaging sensitivity. In this talk, I will be referring to the NTRAC when talking about the product, which are the nanoparticles in this case. 12 partners from different fields shape the consortium, including technological and manufacturing centers, reputed public research institutions, stem cell experts and manufacturers, preclinical CRO, public institution with focus on the relationship between environment and health, regulatory and exploitation and dissemination management. Asphaleon is part of the regulatory work package, which is led by RABM, the National Institute for Public Health and the Environment in the Netherlands. Why is regulatory guidance required and interested in the NTRAC project? The product is still in early development. But why should we think of regulatory issues at that point? Because there are multiple potentially relevant regulations for NTRAC. As a medicinal product, as a medical device, as an advanced therapy, others such as excipients. This regulation may have different information requirements, and they might even involve different regulatory authorities. Considering regulatory issues already in development will help to focus the research by defining the primary purpose of the product and to avoid repeating studies for regulatory information requirements. Therefore, which seems obvious for the benefit of the project, is a challenging task in this particular case, and we will briefly discuss it referring to its classification. Regulatory activities conducted so far include define the main purpose, determine the expected regulatory classification, meeting with authorities such as EMA, ITF, created a regulatory roadmap, meeting with a national competent authority, and celebrated a regulatory workshop. As an ongoing task, we are drafting regulatory documentation to support future clinical trial applications and to publish recommendations on regulatory aspects. Without going into detail in this part of the classification, since the product being developed in this project are the nanoparticles and not a cell-based therapy, it is relevant to put into context the difficulties encountered in the classification, because they have partly led us to take specific regulatory actions. Main possibilities for classification have been discussed internally within the consortium partners and with several stakeholders. Is the NTRAC fitting with the definition of a medicinal product, of a medical device, of other type of products such as diagnostic agents, such as excipient, etc.? And the conclusion is relevant because depending on the classification, different regulatory pathways and requirements may apply. The classification depends on precedents, and in this case there are no precedents, neither guidelines available. Depends on the NTRAC intention of use and depends on the NTRAC components and how they achieve its principal intended mode of action, if it's by pharmacological, metabolic, or immunological action. As there are no similar products market, not clear guidelines for this type of products exist. So we started the following regulatory activities to gain knowledge on the pathways to follow. So, which are the lessons learned from the regulatory process? Let's start with the method followed. Information was gathered through three different channels. Information from consortium partners, meetings with competent authorities, and celebrating a regulatory workshop with experts participants. Let's talk about the first channel. On the premise that regulatory guidance or standard protocols were mostly not available, information from consortium partners on regulatory guidance included discussions on the intended purpose of the nanoparticles, and in particular, the regulatory partners defined an initial roadmap critically assessing aspects of the development related to product quality and preclinical data that needs to be generated in order to label the cells and to use them in future in human administrations. This roadmap, since its initial version, has been adapted over the project execution when new insights were available. Thus, feedback received, for example, from authorities or knowledge gained in the performance of the nanoparticles allow us to adapt and design the plan. For quality control of the NTRAC product, the following protocols may eventually need to be developed that were not available at the beginning. The NTRAC production and characterization for the upscale of nanoparticles needs to be developed. Labeling of the cells with particles which may vary per cell type. Quality criteria for the therapeutic action of cells per cell type also. The second channel was getting feedback from authorities. At the beginning of the project, we requested an ITF with the European Medicines Agency. The Innovation Task Force meeting, as explained in the previous talk, is a free and preliminary consultation with EMA. It provides a forum for early dialogue with applicants on innovative aspects in medicines development. Feedback given is the opinion of the participating members of the ITF and might not represent the official opinion of the European agency. Before the ITF meeting, a briefing dossier and a PowerPoint presentation was prepared, including background information on the composition of the product, manufacturing process, labeling of the cells with the NTRAC nanoparticles, and planned preclinical studies. The main objective of the ITF was to define the regulatory classification of the NTRAC, and the main conclusions during the ITF meeting were, as a general feedback, there was an encouragement for development, as there is a need for this kind of tool for cell therapy development. The NTRAC label and its purpose does not fit well with the EU's definition of either medical device or medicinal product. So, on the regulatory classifications, the opinions during the meeting were divided, which might reflect diverging views between member states. And the ITF experts concluded with the recommendation to us that we should choose the preferred regulatory classification and justify it. It was advised to us as well to consult a national competent authority to seek scientific advice on the strategy for the submission of future clinical trial applications, and especially on which data would be required to that. In summary, classification was not clarified after the ITF meeting, and therefore the definition of preclinical plan to fit with regulatory expectations was still challenging. The feedback from the ITF reinforced then our plans to seek a formal scientific advice. A national competent authority with relevant experience with clinical trials using therapeutic cells was chosen. Answers from the national competent authority were based on questions and information provided by us in a briefing dossier. The procedure is not legally binding because it does not represent an assessment on data, but the consortium prepared a preliminary quality and safety data already available, and this was collected and presented to the agency. The objectives of the meeting were getting feedback in specifications, stability plan, manufacturing process and standards proposed, getting feedback in the preclinical development conducted so far and the planned studies, and getting feedback on the proposal of a clinical design for a first-in-human study. In the meeting with a national competent authority, the main conclusions were that the classification may depend on the company's claim of intended function or indication, but independently of the classification, the applicant, in this case the NTRAC consortium, has to show that the product is safe and reliable. In terms of quality, proposed characterization, list of specifications, etc., were revised and additional recommendations and parameters to be determined were added. In terms of clinical, it was revised and agreed a general proposal for the clinical trial design of a first-in-human study. Importantly, the agency highlighted that results of preclinical studies may impact in the benefit-risk of the clinical trial in patients. In terms of preclinical studies, the following recommendations were given. Optimal ratio particle cells should be determined to correlate the number of cells to the effectiveness in the non-clinical models. More data would be necessary on how to differentiate in vivo between viable and non-viable labeled cells in the animal model. Biodistribution and toxicity data should be presented for both the NTRAC nanoparticles and labeled cells, but the specific data required would depend on whether nanoparticles affect or not the cell's property. Then, a thorough assessment of the cell properties, such as viability, functionality, among others, is key. How long and where the nanoparticles remain in the body should be safe. Nanoparticle accumulation in specific organs should be addressed. And there were also some concerns about the potential interference of nanoparticles with future imaging techniques. It was recommended to additionally conduct an in vivo study comparing the NTRAC nanoparticles and labeled cells against non-labeled cells, because only an in vitro comparability study of label against non-labeled cells might not provide a self-standing basis to assess the impact of the labeling on the biological function of the cells in vivo. And lastly, mathematical and computational models should be validated. It was discussed that the importance on how to assess the risk associated to an incorrect output. Finally, the third method to gather information was the regulatory workshop celebrated. Having in mind to make general recommendations for developers of similar products, a regulatory workshop was organized by our colleagues from RIM, partners of the consortium. We aim to have a forum for open discussion and exchange of experiences, then a multi-stakeholder meeting was done. The workshop consisted in 2 online sessions on 2 consecutive days. Firstly, to disseminate the Engraftment project, and secondly, to obtain expert input on cell labeling. Participants invited were experts from regulatory bodies, academics, companies developing cell therapy products and nanomaterial labels, and also clinicians applying cell therapeutic products. The group was divided into parallel sessions to discuss issues in a more interactive way, and the main conclusions from the sessions were summarized for all attendees in a plenary session. Parallel sessions were focused on the needs and possibilities for labeling therapeutic cells, regulatory classification, quality control and preclinical requirements, and timing of regulatory steps. The main conclusions for the workshop were that the main reason for labeling therapeutic cells is to gain insight into biodistribution and clearance of cells, especially during the development of new cell-based therapy products. This gain information will definitely optimize development and improve treatment options. As a kind of consensus, the different participants agreed that an ideal label system should be inert and ideally compatible with any type of cell therapy. It should also facilitate a number of imaging techniques. Then a multimodal nanoparticle, for example, would fulfill this aspect. And finally, the label should be cleared rapidly from the body. And the last aspect was that the regulatory classification of a nanomaterial label is needed early in the development. However, till now, there was no unambiguous opinion on the classification of NTRAC label. An alternative option from experience in the US suggested to consider the labels as a novel excipient. With regard to non-clinical and preclinical safety data, and more specifically on nanomaterial labels, retention or excretion of nanomaterial label by cells was considered important. Apart from obvious quality criteria of the nanomaterial label itself, quality criteria for each combination of label with cells are needed. For example, concentration of the nanomaterial in the cells, and also cell viability, functionality, once label, etc. Finally, a timeline for critical steps in regulatory process was interactively created in a mural. In one of the parallel sessions using this mural, the participants designed a timeline for the different development phases of a new label system. The timeline, it is basically following the scheme of a new drug development in terms of phases, but with a focus on the specifics of developing systems such as these labels, aiming to label cells. For example, in the prototype selection, it is important to ensure the feasibility of label manufacture and also to ensure proper cell uptake. Therefore, it was specified that reproducible production on lab scale was required, together with a protocol for cell uptake in proof of concept cell models. And here you have some other more details on each single step. And as the workshop driver was regulatory, the group finalized the session identifying the main regulatory actions up to clinical stage. And the main regulatory actions were determining label classifications, identifying applicable standards, talking with regulators in a formal way, and ensuring that preclinical development will enable future clinical trials. Even the actions were focused in an European development, the same applies for other territories, of course, checking perhaps different standards and consulting different stakeholders and different regulatory bodies. As a general conclusions and remarks, there is a high uncertainty about the legal status of a nanomaterial label for cell tracking. Navigation between the tangled requirements of different regulations makes its development challenging. A straightforward roadmap for gathering regulatory required information on nanomaterial labels for cell tracking could save resources. Different objectives of tracking cells in vivo represent several needs. Preclinical development then should be adapted on a case-by-case basis. But independently of the classification of the nanomaterial label, the product has to show that it's safe and reliable. And concluding remarks on preclinical aspects highlighted that the comparison of labeled cells against the non-labeled cells should be done in vitro plus in vivo in a relevant animal model to the targeted disease. The importance of in vivo differentiation between viable and non-viable labeled cells in the animal model is high. It is a very important point for the development of the product to know whether what is observed in the image are indeed viable cells, because the objective of the product is not to track nanoparticles, but to track viable cells in humans. It is also important to be able to correlate the number of cells to the effectiveness in the non-clinical models to justify the positive benefit risk in a clinical study. Biodistribution and toxicity should be presented for both the label and the labeled cells, but the specific data from the labeled cells might depend on whether the label system is affecting or not the cell properties. So, in summary and to conclude, as there is no sufficient guidance for nanomaterial labels to track cells and to be tested clinically, the developer should justify how to test the safety of the label and how the benefit-risk ratio for humans is fully supported. Thank you very much.
Excellent. Thank you very much indeed, Marta. Again, another excellent presentation and run through of the aspects of the trials and the studies that we're really interested in. So, next step now is to move forward to the panel discussion and the live Q&A. So please can I invite speakers to come on to camera if they may, and be ready to come off mute when the questions are directed towards you. So Slido is still open for those who you wish. Here we go. Thank you very much, Lucilia. So slido.com 868602, or scan the barcode on your phone or tablet. It should take you through, and you can type in your question there. You can also, if you scan through the questions, agree or would like to hear the answer to that, vote for questions, and they will then rise up the order list and come to our attention, and we will prioritize those questions. We already have a good number of questions that have been posted. Please do keep adding your questions, and we will start to work through those right now. Okay. Thank you very much. Okay, so our first question I will direct initially to Tineke, but if the other panelists wish to add, please do so. So, how the reason to do preclinical imaging relate to the reasons to do clinical imaging?
Hello, this is Tineke. Actually, until now, I haven't seen so much of clinical or preclinical imaging studies for products in registration phase or development phase. But I can imagine that for preclinical imaging, it's quite difficult if you use the human cells. So for clinical imaging, you will get more relevant information. But for preclinical imaging, one could use an homologous model where the animal equivalent of the cell is used in an animal. Yeah, most of the times I think it is still a quite regarded a quite expensive method to apply preclinically. So that's what I can say about it. Maybe Kate can also comment.
Yeah, so I kind of, this question kind of to me means how does preclinical imaging inform clinical imaging? So one, you want to make sure the cells actually go where you're intending them to go. So in preclinical and clinical non-invasive imaging, you can verify accuracy of cell injection at the target site. And preclinical imaging studies can help inform imaging in the clinical trial. So for example, when injecting cells in specific locations like solid tumors, lymph nodes, or various areas of the brain, you want to make sure the cells are injected accurately into the intended location. So you can do preclinical and clinical imaging depending on the product, route of administration, and the indication. Things like MRI or PET scan can allow you to visualize the cells for both. Other reasons, for preclinical imaging include feasibility, translated to the clinic. Also, data from preclinical imaging can help inform trial design and appropriate monitoring for your proposed clinical trial.
Thank you, Kate. Marta, do you have anything to add to this from your experience?
Yeah, thank you. Well, first of all, I want to say thank you to the CT TRACS for inviting the NTRAC to this meeting, because it really gave us the opportunity to disseminate our project. And I can talk about in the context of the NTRAC, for example, the aim that we were looking for is to generate data first at the preclinical level and then generate algorithms from the preclinical data that help predicting the efficacy of cell therapies as well as biodistribution in the clinical setting. So this data could be then validated at the clinical level. So parts of the things that Kate was commenting, and specifically in our context related to the predicting efficacy and biodistribution.
Excellent, thank you very much. So our next question, thank you very much for voting these up. This really helps us with prioritizing creative questions. So what is the view of the panel on the lack of widely agreed reference standards for in vitro immortalization, insertion-related tumor genicity assays? Perhaps Kate, I could come to you for this one first.
Yeah, so as I mentioned in my presentation, we take a weight-based approach for each product that comes through our office. We look at all of the data and kind of make a decision based on the totality of the submission. So for products that have already been well characterized and maybe have a low tumor genicity risk, the lack of widely agreed reference standards may not be relevant. But again, everything is a case-by-case basis, and we just look at the totality of the submission.
Thank you. Tineke, do you have anything to add to that? It's very thorough.
Yes, this is Tineke again. With regard to widely agreed reference standards for assays to detect in vitro tumorigenicity, I think we put some efforts in that already in CT TRACS. And I think it's valuable because it's good to know if you use an assay to to study in that way whether a product has potency to be tumorigenic. I think you want to know that it's a good assay and that you can trust it, or that you know that it's a sensitive assay. So that's how I understand the question. So in that sense, I would say that it's good to have assays of which we agree that they're valuable to use and to address these risks. I hope this is kind of an answer.
Yeah, absolutely. Thank you very much. And Marta, from your perspective, do you think?
Yeah, in this case, I would
be good.
Yeah, in this case, I would talk as from the developer's point of view. Of course, the lack of reference standards is a further challenge because it really forces its developer to justify their selected and used methods. But on the other hand, considering how different are these type of products compared with classical chemical entities, and that within the HGMPs and cell therapies, etc., there is a broad range of different type of products. It also gives some room to select the most appropriate model, I say, in any case. But from the developer point of view, it's really a further challenge.
Thank you very much. Thank you. So our next question. Clinical imaging has been recommended for some cell therapy products. Can you think of a situation where it may be warranted? And I'll direct that one to Tineke first, if I may.
Okay, this is Tineke again. Can you think of a situation where it may be warranted? I find that actually a difficult one. I would say that sometimes it really supports the pharmacodynamic studies and the pharmacology of the product. And then I would say you cannot see it separate from the pharmacology and from the rationale for clinical treatment. So then it would be really warranted, and to put up a situation that one is maybe a bit difficult, but for one of the CD34 positive cells that was genetically modified with a lentiviral vector for expressing a gene, I would say the PK studies, but that was not an imaging study, in a preclinical dossier, showed that this product also engrafted in the brains, which was very important for its function there to produce the protein locally. And this really supported the pharmacology. And this was, in this case, shown by expressing or having the cells in the animal with an GFP construct expressed. So in a way, it was imaging, it was only not clinical. So in that sense, I should maybe pass the question to Kate or to Marta for an additional opinion.
Okay, yeah, I can jump in. Some situations where clinical imaging may be warranted would be things like novel product types, novel delivery devices, routes of administration, various combination products where, we may be concerned about ectopic tissue formation, distribution in non-target tissue, tumorigenicity potential. If a sponsor is unsure if preclinical imaging is warranted, I recommend reading the preclinical guidance, or like I said in my talk, requesting either an interact or pre-IND meeting, depending on the phase of development, where sponsors can get their specific questions answered.
Thank you.
Yeah, I can jump in now then. Well, in fact, we were discussing this broadly within the consortium because one of the main interests is from the ENTRA point of view is to realize and to know if the clinical imaging is interesting and necessary. And based on so many discussions and conversations with different people also and experts in the field, they reinforce us on the importance and potential need on that, maybe in the context of development of new therapies, as Kate was saying. In the context of new therapies, it can be an interesting tool to select the best one. Of course, if you arrive to the clinical use, it's because you passed all the non-preclinical tests and you have really a lead decide and really want to continue with this one. But even though it can be validated at a clinical level, and also it might help defining the most appropriate clinical dose.
Thank you. Thank you. It just popped into my head here if this question is actually referring to, say, a marketed product, but whether clinical imaging may be recommended for a marketed product. And could we think of a situation may that be warranted? I guess my initial sort of thoughts on this, if we do preclinical and indeed clinical assessments properly, then for a marketed product, we should not have to warrant the need for clinical imaging once it is a marketed product. One would hope.
So I can think of one instance. Yes, it's not. It's a, you know, I'm a pharmacology and toxicology reviewer, so this is kind of way down the road. But you know, say something is marketed and there's some safety signal. You know, we may recommend doing clinical imaging at that point. You know, if we're concerned about that.
Excellent. Thank you very much. Thank you. So our next question is directed to you, Kate. So are nanoparticles for tracking cell products considered under CBER, CDRH, or CBER, CBER? Does it require its own BLA/NDA or for use in cells?
Yeah, so basically they're asking which center has the jurisdiction for nanoparticles for cell tracking products. So this would be a question. We have a pathway. As new technologies emerge, we have a pathway for sponsors to ask these questions to get clarification on where to submit their file. A lot of times this changes as we reorganize, and you know, different centers will then, you know, sort of get different types of products. So I highly recommend, you know, sending a request to ask what avenue to go down for this, because it can change, and it probably depends on, you know, the characteristics of the product. Sometimes different product types are split based on certain characteristics between centers. So I definitely recommend requesting that from FDA.
Lovely. Thank you, Kate. A question directed to you, Marta, please. Could you discuss the most unexpected regulatory hurdle you faced?
Yeah, well, with no doubt, the most relevant regulatory hurdle is the difficulty in classifying the ENTRA product, I mean the non-label. However, this difficulty was not unexpected, especially in the EU territory, where the system of competent authorities includes both EMA and national agencies, and where views, opinions, clinical practices even are not completely harmonized. And additionally, some other bodies can enter into the context, like, for example, notified bodies, etc. So the opinions may also diverge. But what has been unexpected for us is that after successive conversations in different forums with regulators and in informal forums with experts, we were still not sure about which type of product we are developing in terms of product classification. Specifically in the EU, where we know, for example, that the definition of diagnostic agents is under discussion, and it is likely that until there is consensus on this type of products tracking therapies and not diseases could be considered not diagnostic agents, for example. Until this would not be clarified or agreed, maybe we would not be able to have a clear position from the authorities. Fortunately, this limitation has not been an obstacle to obtaining guidance on the data that must be generated in order to use the NTRAC label cells in first-in-human clinical studies. So we are moving forward, but with this difficulty in our hands.
Thank you, Marta. So a question now, which I'll come in turn to. Oh, it jumped. I'll go with this one first, which is: do regulators ask for pharmacokinetics or imaging data specifically? Apologies if that's come slightly out of order. We're jumping around a little bit. So can I come to you, Kate, first, then to Nika, please?
Yeah, so it's extremely variable. You know, every submission is evaluated on a case-by-case basis. Things that might affect whether distribution studies are needed are, you know, characteristics of the product, the route of administration, the indication, the target patient population, and the current state of scientific knowledge for that, you know, particular product type. So again, you know, I always recommend reading the guidance and then, you know, coming in for the meetings, Interact or Pre-IND meetings. With those, we get your entire submission and we can really answer your specific questions based on all of the factors that we look at kind of when deciding if distribution studies are needed.
Thank you, Kate. Nika.
I would say that the new guideline, the ICHS 12, is also quite clear on that. I think in most cases it's valuable, and what we see is that developers do it already quite early in the process, at least the preclinical pharmacokinetic studies. And also what's said in ICHS 12 is that, well, it's not yet an approved guideline, but it's actually now in consultation phase, but it's for hematologic or cells of hematologic origin. It's not always needed, as these are given intravenously and also distributed in the blood. So there it's not so much extra data, but in most other cases, I would say extremely valuable. And we do not request too often because they are most often there already.
Interesting. And Marta, was this a question that you were asked, or had you already got to the point of providing the data?
Yeah, I also have some information that we learned from our process related to that. In the context of the NTRAC project, we were recommended to measure the label cells in animal models over the time, so that it can be proven that the cells with the nano label were clear or not from the body over the time, with a center over the time. So in this case, imaging was useful due to the NTRAC label. So definitely this was specifically recommended to us.
Excellent. Thank you very much. So the next question, would you consider a lack of info on fate of therapeutic cells to be a major bottleneck for regulatory approval? I.e., will imaging increase chances of approval? Nika, I'll come to you first on this one, and then ask for Kate to contribute.
Difficult one, I would say. I think that at least, well, I'm a preclinical assessor, and preclinical is hardly ever a bottleneck, I would say. But it's important to know about the fate of the cells. I mean, if the cells have to have a long-term effect, but not present after 2 days after administration, that really provides no support at all for your mode of action. So and that's the point. It supports really your mode of, or your pharmacodynamic studies and the proof of concept of your product, and it also informs on the risks that are that can be expected. So again, extremely valuable, but if it's really a bottleneck, I doubt.
Thank you. Kate, please.
Yeah, so when thinking about things that are bottlenecks for files that come through, lack of sulfate data is in general not usually the rate limiting factor. There's a huge variety in the quality of submissions that we receive for both, in both pre-INDs and INDs. I would say the most the things that cause the biggest bottleneck are, first of all, just missing data. Either the sponsor didn't submit their complete data, or they just don't have the data because the studies weren't conducted. And I guess for pre-IND meetings, asking the right questions. Sometimes you get one pre-IND meeting with FDA, so you kind of have one chance to ask the relevant questions that will get sort of the most benefit out of the meeting. So really thinking through those and asking the right questions. So I would say those are kind of the biggest bottlenecks, but in general, lack of sulfate data, isn't what we see when we have kind of bottlenecks or major concerns or issues with these files.
That's really interesting. Thank you. Marta, this one of the things, but from your perspective, how much effort or emphasis did you put on for thinking that you do around determining sulfate?
Well, in our case, a lot of efforts, I would say, because we are developing a nanolabel, which is being used in combination with a cell therapy already in development. So one of the important aspects for us is to figure out if the nanolabel is affecting or not the cell's properties and characteristics. So all potential characteristics that can be affected are being assessed as much as possible from our side.
Thank you. So I'm going to sort of jump down a particular question that's been highlighted to me, that there's a question that's sort of closely related to this. So from a regulatory perspective, how is pharmacokinetic and persistence data used or analyzed in the context of assessing cell therapy efficacy in vivo? I don't know if you could pull that question up. Here we go. So this time I'll come to Kate first, if you could give us a...
Sorry, the audio went out I didn't quite catch the question.
It's now highlighted in blue at the top here. So from a regulatory perspective, how is pharmacokinetic and persistence data used or analyzed in context to assessing cell therapy efficacy in vivo?
Yeah, so again, everything's on a case-by-case basis, but yeah, and I mentioned it in my talk a little bit, you can use information that's obtained from distribution studies for, your POC data, for some safety data. When feasible, we definitely recommend hybrid studies where you can get data for all three in one. That's really great when there's an appropriate disease model available, and it's judicious use of animals, and you can kind of get all that data in one large pivotal study. Some things that biodistribution data can inform for safety data or POC data would be, dosing regimen, dose levels, things like that. So there's really a lot of information that can be sort of, that can overlap between biodistribution studies and safety studies.
Excellent, thank you. Seneca, please.
I'm not sure whether I get the question right, but I would say that most of the times I see that the persistence data, as well as, or the pharmacokinetic and the persistence data, is retrieved from one study and then related to the efficacy data from a pharmacodynamic studies. So one supports the other, but maybe sometimes these are combined as well. I have at the moment not an example in my head about that, but I mean, the principles, if you know about the persistence of the cells, it supports your efficacy, I would say.
And thank you. And Marta, using the technology that you described in your study, can you give us an indication of how that might go towards providing some of this data?
Yeah, in fact, the technology that we are developing would help in providing additional data that could be reinforce the pharmacokinetics in terms of imaging, biodistribution, etc., and even persistence, because the image can stay for a long time or not, depending on the clearance. So. Yeah, we think that this a tool like NTRAC could help in providing this kind of data that in future could be assessed by regulators in the context of an application. Yeah, but we will see.
Okay, thank you very much. So one more question, and then we'll have our final wrap-up question. So the question that's at the top of the list now here is, so what weight, related, I guess, what weight is put on biodistribution studies when reviewing an application? And let's come to you first, Kate, please. Last time.
Yeah, so I kind of feel like a broken record here, but it's tough to make a generalized statement on that because it's just so product dependent. From my talk, you saw that slide. We just have such a wide variety of products that come through the office. For maybe for one product, it's extremely important, and we will put an IND on hold, without distribution data because we're that concerned about what might be happening. For another product where we've seen that product type many, many times, we have a good scientific knowledge of it. It's a very common route of administration, a common dose level, a common dosing regimen. We may not need it at all. And so it's, and then there's everything in between. So, all of those factors play in. And again, really, I think there's a lot of mistakes that sponsors make that potentially can be avoided by really reading through the preclinical guidance that's available online, and then also taking advantage of the opportunity to come and have a chat with us about your specific product, your route of administration, your indication, and we'll give you directed specific feedback on kind of what you need to do to have a successful IND.
Excellent. Tineke. Oh, I think we're going to move on to our.
I'm sorry, wrong moves. Given the time, yes, I think we should move to the closing question. So I'll put that one on the screen.
Thank you very much. Yeah, how time flies. That last 30 minutes has really raced by. So our closing question, really just briefly to each of the participants, and Marta, I'll come to you first. What do you see coming in the horizon in terms of safety assessment of cell therapies, potentially new methods, gaps, or needs? And what do you see as the hope for cell new therapies in the future?
Not my now.
There we go. Oh. Now.
Okay, I couldn't unmute myself. Yeah, what I would like to see in future solutions like the NTRAC, of course. This is why we are working on this project and developing a tool like this one. I think it's important to follow new cell therapies, not only from a safety point of view, which of course is crucial, but also from an efficacy point of view. So this is why different solutions such as NTRAC could be useful, and this is why we are pursuing this objective within the project.
Thank you very much. Kate, I'll come to you next, please.
Yeah, so there's a lot of exciting things going on in the field right now. Marta had a wonderful talk on NTRAC. I just watched a talk over the weekend on a new technique of label-free tracking of unlabeled stem cells, which was pretty neat. Obviously without any cellular modifications, you don't need to worry about changes to, cell viability, phenotype, or activity. I'm always looking at what's happening on the in vitro side with tissue engineering to assess activity, distribution, and safety of cell and gene therapy products to reduce animal use. And it's always exciting to watch these cell therapy products move through the clinics, especially when we see an encouraging safety and efficacy data come out, because at the end of the day, our main goal at FDA is public health and, trying to get safe, efficacious products out to the people who will benefit from them. Thank you.
Thank you, Kate. Tineke, please.
Yes, I agree with Kate that the in vitro options are growing, and we look forward to have validated models to check these things out, to the pharmacology and toxicity of the cell therapies. With regard to the biodistribution, I'm looking forward to see more examples of including the biodistribution in the clinical studies via imaging. And with regard to safety, I think that integration site analysis will become even more important than it's right now, because we have some studies put on hold because of signals of tumorigenicity, and it may be not due to the therapy or not due to the integration of a vector, but I think there are some uncertainties about these issues. So we will see more about that, I think. Yeah, thank you.
Thank you very much. So we are out of time, so I won't do a wrap up. I think it's in terms of summary of what we've done. Just from my perspective, listening to the talks and the presentations here, and also the questions, the approach to get these therapies to market, to get these therapies through these processes, is clearly collaborative, and that's from all sides: therapy developers, the regulators, and the technology and tool developers. And it's wonderful to see that, and long may that continue. So just in final, to round up with some thank yous. So to the Artris team, to Courtney, Perrette, and David, thank you very much for that. For the team at HESI TRACS with Lucilia, who's driven this forward, and Connie in support. And of course, and contact details are on the slide now. And of course, to our speakers, to Marta, to Kate, and to Tineke, the effort to put these presentations together and the pre-recording and preparations are not small. So many thanks to you all. And finally, to all of you for joining us and listening. But without an audience, there'd be no webinar. And thank you very much indeed for all of your questions and your comments. And we very much look forward to talking to you at an event in the near future. If there's nothing else, Lucilia, no further comments, questions? I think we're good. Thank you very much indeed.
Thank you all.
Thank you very much.
Thank you. Have a lovely evening
Thank
or rest
you.
of your day. Thank you.
Bye.
Thank you.